Welcome to the Deep Dive Lab, where we dive deep into fascinating science stories filled with surprising discoveries, unexpected twists, and thought-provoking ideas.
Thanks for having me back.
Of course. So, to start us off today, I want you to imagine a factory. And this factory produces a highly volatile, incredibly essential fuel.
Right. A fuel the whole system absolutely needs.
Exactly. If this facility doesn't make enough of this specific fuel, the entire surrounding infrastructure just shuts down. Like, we're talking complete systemic blackout.
Which is bad.
Very bad. But there is a massive inherent risk here, too, because if the factory overproduces this fuel, the product itself becomes highly toxic.
Yeah, it basically turns on its own environment.
Right. It accumulates and literally burns the factory down from the inside. So the big question is, how does a system like that know exactly when to hit the kill switch on its own assembly line, before the fuel reaches that critical toxic mass?
It is honestly the ultimate biochemical balancing act. And I mean, this isn't just some abstract thought experiment for you to ponder. This exact scenario is literally happening inside your cells right now.
Now. Wow.
Yeah. The fuel we're discussing is heme, that crucial iron-coordinating molecule that carries oxygen in our red blood cells.
What makes blood red, right?
Exactly. And it's essential for cellular respiration. So the assembly line worker endlessly pumping out the precursors for this heme is an enzyme called ALAS.
ALAS, or aminolevulinic acid synthase, for the biochemistry enthusiasts out there.
Good memory. Yeah, it's the gatekeeper. It represents the very first rate-limiting step in the whole heme production pathway.
So if ALAS is active, the assembly line is moving.
Right. Which brings us to the core of our deep dive today. We're looking at this really remarkable piece of biological detective work.
Yeah, published in July 2026 in the journal Science, right?
That's the one. A team of researchers from Brandeis University, Boston Children's Hospital, and Harvard Medical School set out to uncover a missing mechanism.
Because, for decades, we've known the body must have a way to stop heme production when levels get too high.
Right, to prevent that toxicity you mentioned. But the actual trigger, that microscopic kill switch, was completely unknown.
Okay, so where did they even start looking?
Well, we did know about the factory supervisor, at least. Inside the mitochondria, there's this highly sophisticated protein-degrading machine called CLPXP.
CLPXP, okay.
Yeah, you can think of it as a molecular shredder. And under baseline conditions, CLPXP actually supports the ALS enzyme.
Wait, a shredder supports it? How does that work?
It partially unfolds ALS. It essentially massages its molecular structure just enough so it can bind to a necessary cofactor.
Oh, wow. So it helps LS do its job more efficiently.
Exactly. It's facilitating production. But when heme levels cross that critical threshold, the dynamic completely flips.
Here comes the kill switch.
Right. This supportive molecular machine turns into a full-on destructor. CLPXP stops assisting LS and instead completely shreds the enzyme.
Taking it offline and halting heme production entirely.
Exactly.
So if we look at this from just a logical thermodynamic perspective, the most elegant mechanism would just be direct feedback inhibition, right?
That's what you would think, yeah.
Like the excess heme would simply bind directly to the CLPXP shredder, induce a conformational change, and just boom, shift it from support mode to destroy mode.
Right, and that was the prevailing hypothesis for a very long time. I mean, it makes perfect evolutionary sense to have a direct two-component feedback loop.
The signal interacts directly with the machine.
Exactly. But biology often refuses to be that simple. When researchers finally managed to isolate human CLPXP and the ALAS enzyme and put them together in a test tube,
the elegant theory just fell apart.
Completely fell apart. They flooded the test tube with heme, expecting to watch this shredder go to work on ALS, and nothing happened.
Wait, really? Nothing at all?
Absolutely nothing. The degradation machinery was completely blind to the heme.
That is so weird.
I know. The ALAS enzyme remained perfectly intact, despite the environment being completely saturated with the exact signal that was supposed to trigger its destruction.
So the heme is just floating there and the shredder is just staring at it, doing nothing.
Right. This is what we call a reconstitution failure. And it usually means you are missing a piece of the puzzle. There had to be an unseen middleman, a distinct adapter protein that senses the heme and physically bridges the gap between the ALAS enzyme and the CLPXP shredder.
Which sets up the central mystery the team was trying to solve. They were hunting for a ghost in the mitochondrial machinery.
Exactly. Led by researcher Julia Harden, they started looking for this phantom adapter.
And when they finally identified a prime suspect, it was honestly incredibly perplexing.
Very much so.
The protein they found is called PULDIP2, And when I was reviewing the source material, this jumped out at me immediately.
Because of what it normally does.
Yes, because POLDIP2 polymerase delta interacting protein 2 is famous for being a nuclear protein.
Right, it hangs out in the nucleus.
Its well-documented day job is assisting with DNA replication. That's like finding out the company's IT guy is secretly moonlighting as the executioner on the factory floor.
That is a perfect analogy.
What is a DNA replication accessory protein doing inside the mitochondria regulating heme synthesis?
It's a staggering biological pivot, honestly. Researchers had noted some faint signals in the past suggesting POLDIP2 might have a mitochondrial isoform, meaning a version of it that gets shipped to the mitochondria instead of the nucleus, but no one knew what it was actually doing down there.
Until the Cardin team decided to test it.
Right. They took that failed test tube experiment, the one with the CLPXP shredder, the ALAS enzyme, and the heme, and they introduced purified POLDIP2 into the mix.
And boom, that's the breakthrough.
Exactly. The introduction of POLDIP2 immediately activated the CLPXP shredder, and ALAS was rapidly degraded.
POLDIP2 was the missing bounty hunter.
Yes. But the key detail here is that POLDIP2 only facilitates this destruction when excess heme is present.
Oh, interesting. So if you remove the heme from the test tube,
POLDIP2 completely ignores ALAS. It is a strictly heme-dependent mechanism.
Which brings up a fascinating structural question, I think, because proteins are these incredibly complex three-dimensional shapes, right? Folded in very specific ways.
Highly specific.
So for POLY-T2 to act as a bridge, it has to physically bind to ALAS, but only when heme is in the vicinity. How does a small molecule like heme suddenly cause two massive proteins to stick together with such high affinity?
That was the next big hurdle. How is this actually working mechanically?
Yeah, because I initially assumed it was standard allosteric regulation. You know, heme binds to a pocket on POLY-P2, changes its overall shape, and that new shape just happens to fit perfectly into a groove on ALAS.
Right, like a key turning in a lock, changing the shape of the mechanism.
Exactly. That's how a lot of these cellular signaling pathways work.
It's the standard textbook mechanism, for sure. But to find out if that was happening here, the research team utilized AlphaFold.
The AI system.
Yeah, the AI that predicts 3D protein structures from amino acid sequences. They modeled the interaction between POLDIP2 and ALAS in the presence of heme.
Okay, and what did it show?
What AlphaFold predicted, and what the team subsequently proved, was far more direct than allosteric shapeshifting. The heme molecule wasn't just triggering a structural change, it was acting as physical structural glue.
Wow. Okay, let's dive into the methodology of how they actually prove that, because you can't just rely on an AI model, right? You need empirical validation.
Absolutely. So the researchers used UVV spectroscopy.
Oh, okay.
This is a technique where you shine ultraviolet and visible light through a sample and measure exactly which wavelengths get absorbed.
Because molecules absorb light differently depending on how their electrons are shared, right?
Exactly. It gives you a highly specific signature of molecular bonds. And heme has a very distinct iron atom right at its center.
Right.
The way that iron atom coordinates with other molecules dramatically shifts its light absorption signature. So the researchers looked closely at the amino acid sequences of both proteins.
And what did they find?
They found that ALAS has a highly conserved sequence, a cysteine-proline motif, and POLDIP2 has a specific amino acid as well, a histidine at position 225.
Okay, so based on the AI modeling and the spectroscopy data, they realized that the cysteine from the ALAS enzyme and the histidine from the POLDIP2 protein were both reaching into the center of the heme molecule.
Yes, they were physically co-coordinating that single shared iron atom.
That is mind-blowing. They are quite literally sharing the iron atom to form a single, unified structure.
They are, and the UVV spectroscopy confirmed this beautifully. When both proteins bind to the heme simultaneously, the shared electron environment around that iron atom shifts the light absorbance.
Creating a specific peak, I assume.
A massive, highly specific peak at exactly 425 nanometers. It's a spectral fingerprint that only exists when all three components, ALAS, POLDIP2, and heme, are locked together.
That is an incredible visual. I mean, it's not just a chemical signal floating by and flipping a switch. It is a literal structural bridge.
Exactly. Without the heme molecule acting as the central anchor, the two proteins simply cannot hold on to each other.
Because they need that shared iron atom.
Right. And if the researchers mutated either the cysteine on LAS or the histidine on POLDIP2, the 425 nanometer peak completely vanished and the degradation process halted entirely.
Wow. It reveals such an elegant economy in the cell's design, you know. The product of the pathway heme is the literal physical component required to dismantle the pathway's creator.
It's poetic, almost.
It really is. Okay, so we've established how POLDIP2 recognizes LS and uses heme to lock onto it. But I see a mechanical issue here regarding the actual destruction process.
Oh, what's that?
Well, the CLPXP complex is a hexameric ring, right? It looks like a microscopic barrel.
Yes, that's a good description.
To degrade a protein, it has to grab a loose end of the amino acid chain and forcibly pull it through the narrow pore of the barrel, unraveling the 3D structure as it goes.
Right.
So if POLDIP2 is essentially hugging LLS, how does that actually help CLPXP feed it into the shredder?
You're touching on a fundamental principle of biochemistry here, known as the directionality of protein degradation. You cannot just push a folded protein against the side of the CLPXP barrel and expect it to break down.
Right, because it requires threading a needle. The protein must be fed into the pore from a specific end.
Exactly. An amino acid chain has two distinct ends. The N-terminus, which is essentially the head where synthesis started, and the C-terminus, which is the tail end.
Okay, head and tail. Got it.
Through their structural analysis, the researchers discovered that the molecular glue connection, where POLDIP2 and heme bind to LLA, happens exclusively at the N-terminus, the head of the enzyme.
So POLDIP2 locks onto the head.
But here's the critical twist. The CLTXP shredder cannot initiate degradation from the N-terminus.
Wait, what?
Yeah. To unravel OLS, the shredding machinery must grab the opposite end. It has to pull the C-terminus or the tail.
Wait, so the adapter protein is holding the head, but the destruction machine requires the tail. How does holding one end facilitate the machine grabbing the other?
The researchers demonstrated this through a beautifully elegant genetic experiment. In the lab, they truncated the ALAS enzyme. They genetically snipped off the C-terminus, removing the tail completely.
Okay, snipping the tail off.
When they ran the degradation assay again, PLDIP2 still used the heme glue to bind perfectly to the N-terminus head. The connection was flawless.
Because the head was still there.
Exactly. But the CLPXP shredder couldn't do anything. Without the tail to pull on, the protein could not be degraded.
Oh, wow. That completely redefines what PLDIP2 is actually doing. It's not just handing a last to the shredder. It's acting as a spatial anchor.
A spatial anchor. That's a great way to put it.
It's like PLDIP2 grabs the worker by the collar to hold them still purely so the CLPXT shredder can grab a loose thread on their pants and unravel the whole sweater from the bottom up.
That is a very vivid image, but yes, mechanically that is exactly what has happened. By tightly gripping the head of the enzyme, PLDIP2 restricts the molecule's movement and physically positions that flexible tail so that the CLPXP barrel can easily grab it and start pulling.
It sets the directionality of unfolding.
Yes. Both the anchor point at the head and the loose thread at the tail are absolutely mandatory for the execution to occur.
It highlights the sheer mechanical precision required at the molecular level. It's a choreographed sequence, right? The accumulation of heme triggers the binding at the head.
Which stabilizes the structure just enough for the shredder to catch the tail.
And pull the entire enzyme apart, amino acid by amino acid.
Exactly.
Okay, this level of mechanical detail is fascinating, but we need to ground this in human reality for a second. So what does this incredibly specific molecular dance mean for me or for human health?
Well, when a system this precise breaks down, the consequences are usually severe. This specific pathway and the failure of this pathway is directly linked to a debilitating human disease called X-linked protoporphyria, or XLPP.
XLPP, I've heard of porphyrias before, but this one is particularly severe, right?
It is a very severe condition. Patients with this disease experience a massive systemic buildup of a molecule called protoporphyrin IX, often abbreviated as PPIX.
And PPIX is the immediate precursor to heme, right? It's
Yeah.
the molecule that is just waiting for that final iron atom to be inserted.
Exactly. When the ALAS factory runs out of control, it produces vast quantities of PPIX. The problem is that PPIX is highly phototoxic.
Phototoxic, like reactive to light.
Incredibly reactive. When it accumulates in the bloodstream and tissues, even brief exposure to sunlight causes severe burning pain in the patient's skin.
Oh, God, that sounds awful.
It is. And in the most severe manifestations, the accumulation leads to catastrophic liver damage, sometimes requiring a full liver transplant.
Wow. And for years, the medical community understood the genetic basis of XLPP, right? Like they knew it was caused by a mutation in the ALAS gene.
They did. Specifically, the mutation causes a frame shift that alters and truncates the C-terminus of the enzyme.
The tail?
Yes, the tail. So patients with XLPP are producing ALAS enzymes that are missing their tails.
But prior to this research from Brandeis and Harvard, the assumption was simply that losing the tail altered the enzyme's internal structure and made it hyperactive.
Right, it was thought to be a gain-of-function mutation, where the enzyme just worked too fast. But this new discovery entirely rewrites that pathology.
Because it's not hyperactive.
No, the enzyme isn't necessarily hyperactive, it's simply immortal.
Wow, because it's missing the tail. The excess heme is still present. PLDAP2 is still doing its job, using the molecular glue to lock onto the N-terminus head.
But when the CLPXP shredder reaches for the tail to unravel it, there's nothing to grab.
So the factory worker goes rogue, cannot be fired by PLDAP2, and just endlessly pumps out toxic precursors.
Exactly. The degradation fails, the enzyme remains active indefinitely, and the toxic PPIX continues to flood the system. To definitively prove this entire feedback loop, the researchers took their findings into a living model.
Mouse models, I assume.
Yes, they utilized CRISPR-Cas9 gene editing on mice. But instead of mutating the ALF enzyme to recreate the human disease, they targeted the newly discovered adapter.
They deleted TLBPO.
They completely deleted the POLDP2 gene in the red blood cells of these mice.
Okay, I have to pause you there because that raises a massive red flag in my head.
Oh.
If they use CRISPR to knock out POLDIP2, a protein we've already established is vital for DNA replication in the nucleus, wouldn't those cells just undergo catastrophic DNA damage and die long before any heme precursors could accumulate? Like, how do you even measure PPIX levels in a cell that can't replicate its DNA?
That is a brilliant point, and it's precisely why the researchers focused exclusively on mature red blood cells.
Oh, wait, because mature red blood cells don't have nuclei.
Exactly. Mammalian red blood cells are unique. During their maturation process, they eject their nucleus entirely. They don't have DNA, and they don't divide.
So they don't require the nuclear DNA replicating functions of POLDIP2 at all.
In a mature red blood cell, POLDIP2's only remaining job is in the mitochondria, regulating this heme pathway.
That is an incredibly elegant experimental design. So by knocking out PLDP2 specifically in that context, they could isolate its mitochondrial function without triggering cell death from DNA replication failure.
Exactly.
And what happened to the mice?
The results were stark. By simply removing the POLDIP2 adapter, effectively removing the ability of the shredder to recognize LOAS, the mice exhibited a 10-fold explosion in toxic PPIX levels.
Tenfold, just from removing that single bridging protein.
Deleting the adapter protein perfectly phenocopied the devastating biochemical effects of human XLPP. The system completely crashed without its heme sensor.
That profoundly changes how we view diseases like XLPP and other related porphyrias. We aren't just looking at a hyperactive enzyme anymore. We are looking at a failure of targeted degradation.
Right, and that opens up entirely new avenues for therapeutics. Instead of trying to invent a drug that globally blocks the ALSS enzyme, which could be dangerous since we still need some heme pharmacologists, could potentially look for ways to modulate POLDIP2.
It provides a highly specific molecular target. Like, if researchers can develop compounds that stabilize the interaction between POLDIP2 and the truncated ALS in XLPP patients,
or perhaps find a way to recruit a different degradation pathway entirely. It could offer a targeted intervention where currently there are very few options. It moves us from a general misunderstanding of the pathology to a precise structural roadmap of the breakdown.
it is a remarkable progression from a baffling test tube failure to a potential blueprint for treating a severe genetic disorder. As we synthesize everything we've uncovered today, let's distill this research down to 3 critical takeaways for you to keep in mind.
Absolutely. Number 1, the long-standing mystery of how heme limits its own production is solved by PLDIP2. It is the essential, previously unrecognized adapter protein that senses heme and bridges the gap between the ALase enzyme and the CLPXP destruction machinery.
Such a huge find.
Number 2, the mechanism of this connection is incredibly unique. Heme doesn't just send a chemical signal, it acts as a physical molecular glue. ALAS and POLDIP2 physically bind by coordinating a single, shared iron atom at the center of the heme molecule.
But the spectroscopic of 425 nanometer peak, just brilliant. And number 3.
This process requires precise spatial orientation. POLDIP2 anchors the head of the enzyme so the shredder can grab the tail. In diseases like XLPP, a genetic mutation removes that tail. making the toxic accumulation of heme precursors absolutely unstoppable.
It's a masterclass in how complex and interdependent our cellular machinery really is. But this discovery leaves me with a lingering, somewhat unsettling thought for you to explore on your own.
What's that?
Well, we spent decades observing POLDAP2, right? Confidently categorizing it as a well-understood nuclear protein responsible for DNA replication. We thought we had it fully mapped out.
We really did.
Yet all this time, a fraction of those proteins were commuting to the mitochondria, moonlighting as heme-sensing executioners. It forces us to ask, how many other well-understood proteins in our biology textbooks are secretly living double lives?
That is a wild thought.
How many other unexplained diseases are waiting for us to simply look at an old protein in a completely new cellular neighborhood? Thank you for joining us on this deep dive. It is always a privilege to unpack the microscopic marvels that keep us alive and to realize how much of our own biology remains undiscovered terrain. If you found this journey as fascinating as we did, we warmly invite you to follow and subscribe for more scientific explorations. Until next time, keep wondering.